Let me start by saying that, yeah, it is a target-based drug discovery approach that I'm going to talk to you about today. And probably just to make the argument that we probably do need new antifungals, I've put up probably something that's a little bit controversial by saying that drugs don't work brilliantly. They work pretty well, actually, in most cases, and the recent classes of drugs, the candins and the next generation azoles, have certainly improved matters quite a lot. Recent studies suggesting 90-day survival rates are improving, but still poor for candida and molds. Some of this is clearly down to our inability to diagnose these diseases adequately, but part of it as well is to do with the agents that we're using being not particularly effective. And there is worrying evidence, although it isn't extreme at the moment, of multidrug-resistant Candida. And the National Aspergillosis Centre in Manchester has been reporting that 20% of its Aspergillus isolates are resistant to at least one azole. And in the Netherlands, of course, we're seeing very high levels of azole resistance in certain centres, so much so that people are suggesting that we move away from azoles as primary therapy, or at least sole therapy. And in those patients where we see resistant isolates, we're seeing that mortality exceeds 88%, 90%, that sort of level. So therefore, I'd argue that there's probably a decent need for antifungal drugs. What I'm not going to do is ask you the question whether we need new antifungal drugs, because if you say no, that's the end of my talk. So I'll move straight on. So how do we identify a suitable anti-infective drug target? Well, clearly, this is the dogma. A target must be critical for viability of the pathogen. But with which pathogen are we talking about? What are the critical pathogens in terms of fungi that we need to look at? Clearly, there needs to be a clinical need, but there also needs to be a thought about the economics of drug development. And when we talk about the viability of the pathogen, what are we talking about in terms of viability? And I'll touch on that in a little bit later. So our targets have got to be sufficiently different from our host, of course, because we want to kill the pathogen and not the host, so we want to give ourselves the best opportunity of doing that. And this is critical. The target must be druggable. Being druggable, it's a terrible word, but what it means is, can we get the drug to the place where the target is? And when we talk about the target, we're talking about a protein that's carrying out a function. And it's usually inside the cell, inside the fungal cell. So can we get the drug through into the fungal cell to the place where the target is and disrupt the function of that target? That's actually a difficult task. And the other thing is, once we've got our target, can we develop assays to monitor the function of that target? Because if we can, then we can do some high-throughput screening to identify novel drugs and use that as a process to kickstart our drug discovery process. And the other question is, do we have some structural information about the target? Do we know a little bit about what the target looks like? Can we do some in silico work, some computer-based work to try and dock drugs into the target to predict what drugs might be able to inhibit its function? If we do that, it's going to speed up the process of drug discovery quite rapidly. So going back to which fungi we need to target, clearly this paper was published at the end of 2012, suggesting that these are the key pathogens that we need to be looking at. Of course, some of these are much more difficult to treat than others. So Rhizopus, for example, exceptionally difficult to treat with current therapies. But unfortunately, it's not a commercially viable way to go to develop a drug that really just targets Rhizopus. It'd be great if we could develop a broad spectrum antifungal that targeted all our fungal pathogens. But actually, to date, we haven't been able to do that even with what we've got. So it's probably unrealistic to think that on the horizon is a single drug that's going to kill all our fungi. So maybe it's going to be a little bit easier to develop a narrower spectrum agent, and probably our key targets are going to be the ones that are causing the most problem, especially in terms of resistance as well. So when we look at our gene targets, what we're looking for, well, typically with gene targets that we want to target for therapeutic purposes, these targets are essential for biological viability of the pathogen. And when we talk about biological viability, we're talking about in vitro biological viability, because that's where most of our studies have been done. If we look at what has been done in fungi, we've got some really good understanding of what genes are critical for viability in fungi. In baker's yeast, we know about 13,000 genes that are essential for viability, so 13,000 potential drug targets. And this comes from a whole genome knockout study, so they've gone on and knocked out each gene in baker's yeast. And similar studies in, another non-pathogen, but filamentous fungus, Neurospora crassa, another 16,000 targets to go at. It's been a little bit more difficult to identify the essential genes in pathogens, although some headway has been made. Terry Romer and Merck, they identified over 550 essential genes in their study. They called it GRACE. It's quite a complex system, but elegant to identify essential genes. The main difficulty is, of course, that Candida albicans is an obligate diploid, so identifying essential genes through standard process was difficult. And we have identified over 116 essential genes in Aspergillus fumigatus. So we know a lot about essential genes. But in terms of druggable targets, we know a lot less. So we can see, this is an Aspergillus cell, a nice cartoon of an Aspergillus cell. And we can see that most of the drugs that we have available to us clinically are targeting cellular integrity. They're looking at hitting the cell wall or the cell membrane directly or indirectly through its biosynthesis. Although a number of other targets have been identified, actually they've not really been extensively studied, and it's usually just one-off drugs that hit these regions. So, we probably need to investigate the druggable space a little bit better. So we have a lot of information about drug targets, potential drug targets, about essential genes. So we can take a step back now and have a look, see what we think about the biology of these organisms and what we think will be important for their pathogenicity. And one class of potential drug targets that we were particularly interested in because of their biological function was this set of enzymes called phosphopantothenial transferases. So a great long name, but I'll try and call them PPTases. And there are a number of filamentous fungi. In fact, there are 3 different phosphopantothenial transferases in filamentous fungi. They catalyze this enzymatic reaction. This is coenzyme A, and this is a carrier protein. So the function of these PPTases is to transfer the phosphopantothenial group from coenzyme A, which is this bit at the end, from coenzyme A, and attach it to a carrier protein. So that's what's happened here. And what it essentially does for this carrier protein is provide it with this reactive thiol group at the end that allows it then to act as a tether for other reactions to take place. So it's actually, it's an intermediate carrier, this carrier protein, and the PPTase basically charges it for acceptance of building blocks for other things. So what is it involved in? So, as I said, we have 3 phosphopantothenial transferases. One is actually a component of a much larger enzyme. It's a fatty acid synthase involved in production of fatty acids. Critical component of fungal biology and essential for Aspergillus to grow and many other organisms as well. The second phosphopantothenial transferase is also involved in lipid biosynthesis, lipoic acid specifically, but it's the mitochondrial equivalent. So it makes the fatty acids in mitochondria. And we can see this here. We've coupled PPTB to the green fluorescent protein, and we can see that it matches to our dye, which binds to mitochondria. So it's a mitochondrial target, as we expect. And then there's this third class of enzymes that we're particularly interested in and I want to talk to you about today. And they are the PPTA, class 2 PPTases, just to make it confusing. and they are cytosolic, and they have a range of functions. And I'll go on to talk to you about them now. So, diverse role. PPTA activates, as I said, a number of different molecules. Activates a gene or an enzyme called aminoadipate reductase. Aminoadipate reductase is critical for lysine biosynthesis in fungi. It also activates genes called non-ribosomal, or proteins called non-ribosomal peptide synthases. Two key examples of that are SID-C and SID-D. These proteins are involved in the production of siderophores. Now, as you've heard before, earlier in the talks today, siderophores are used for iron acquisition. So they're critical for filamentous fungi, Aspergillus in particular, to sequester iron from its environment. And PPTA also activates polyketide synthases. One example is PKSP, which is involved in the biosynthesis of melanin, the green color that we see in Aspergillus spores. And not only that, but it activates all the other polyketide synthases and non-ribosomal synthases in Aspergillus, of which there are a number. So what happens? So again, we're molecular biologists, we like to knock things out, right? So we knock out the PPTA and see what happens. So this is our wild-type strain, A1160. We can see this green coloration. If we knock out PPTA, if we don't supplement with anything, it doesn't grow. Because our expectation is it needs iron and it needs lysine. So we need to feed it. So if we feed it only iron, it doesn't grow. If we feed it lysine on its own, it doesn't grow. But if we feed both, it begins to grow. But it's white. It can't produce melanin. Okay, so this gene is doing exactly what we expect it to do. And also I said that PPTA activates all the other non-ribosomal peptide synthases and polyketide synthases. So we lack any secondary metabolism, secondary metabolites in Aspergillus. So this is an example of a culture extract from well, actually this is just the media on its own. This is what a culture extract looks like. So we can see various peaks which represent different secondary metabolites, including critically gliotoxin. So gliotoxin is quite a nasty toxin that's produced by Aspergillus, and it's also an immunomodulator. Yeah, it's also critical for Aspergillus pathogenicity. If we knock out PPTA, we get rid of all these secondary metabolites. If we put it back in again, they're restored. Great. And not surprisingly, when we knock out PPTA, because of its requirements for iron and lysine and the lack of secondary metabolism, it's avirulent. So we can see here on the top line, so this is our PPT knockout strain. It's unable to cause any mortality, whereas our wild-type strain kills all our mice within 9 days. Similarly, in a systemic model of infection, all our mice with our wild-type strains are killed within 2 days, whereas our PPTA knockout is avirulent. So it's looking good. So why is our PPTA knockout, our PPTA null mutant, avirulent? Well, we look at the requirement for this mutant for lysine. So as you can see here, this is our wild-type strain again, grown in the presence of various amounts of lysine in an iron-rich media. And we can see that for our wild type, it's able to grow without any supplementation, so it has the ability to synthesize lysine. But our knockout begins to lose the ability to grow around about here, not particularly well here, and then by the time you get down here, it's not growing very well at all. So what do we know about lysine in our host? So this is published estimate, a bit of range here actually, but of the amount of lysine that is in blood. So we can see here we're down to levels which would suggest that Aspergillus would be unable to grow very well. So great. This suggests, on the face of it, that maybe lysine biosynthesis could be involved in the lack of virulence. And indeed, a previous publication suggests that if we knock out lyse F, we attenuate virulence for Aspergillus. Actually, if you look in more detail, and these guys actually did a great job in doing that, they published a follow-up paper that said, actually, yeah, lysine biosynthesis is important in certain tissues, but in an established infection, what seems to be the case is because Aspergillus produces proteases, it's able to break down its local environment and acquire lysine from its environment. So maybe lysine itself isn't the best target in the world. So what about iron? So iron, exactly the same experiments with reduced actually TAF-C, which is a siderophore in this case, but it's equivalent to iron. So we can see that at this level here, this is when our mutant stops growing, and the levels of free iron in blood are exceptionally low, way below these levels. So it may be that this is a useful target. And indeed, yeah, we see studies in the literature that suggest that actually strains lacking the ability to produce siderophores are avirulent. So yes, this is a really useful target. So what about the useful spectrum of activity of this drug target? So we've said that maybe that iron is the key factor here. But also let's have a look at the interrelatedness of the genes in Candida, in Cryptococcus, in Aspergillus. We can see that actually these are the filamentous fungi, essentially, all relatively closely related. And here, out on a limb where there's Candida up here somewhere, that's up there, and then Cryptococcus is around here somewhere as well. So they're actually quite distinct, the phenotype, phylogenetically distinct. So maybe if we're targeting an Aspergillus at PPTAs, we might not be hitting the Candida one. And indeed, breaking up these two groups of fungi, quite surprisingly, is the human enzyme. So there is a human enzyme, and that too is involved in lysine biosynthesis. So the chances of us hitting filamentous fungi and yeast but not human, probably slim. So maybe we're looking at a filamentous fungal specific target. Okay, that's okay. And also, we know with Candida, well, filamentous fungal specific or Candida specific, I suppose one way or the other. But we know that lysine, the ability to produce lysine is dispensable for virulence in Candida. And this is another publication about siderophore biosynthesis, essentially. The genes involved in siderophore biosynthesis. We can see in filamentous fungi, they produce siderophore. They've got genes involved in siderophore biosynthesis. But Candida here doesn't have any siderophore biosynthetic genes. It doesn't use siderophores for acquiring iron. So maybe we need to be looking very much at a filamentous fungal specific drug if we're targeting PPTA. But again, that's okay. So we have our validated gene, essential gene. We know it's essential not only in vitro, but also in a mouse model. But how are we going to develop a drug to target this gene? So we look back at what this enzyme does. It takes this group here and transfers it onto this protein here. How can we monitor this action? And the way we do it is use an assay technology called fluorescence polarization. The way that fluorescence polarization works is it looks at the spin of a molecule. It looks at how quickly it rotates. So what we've done is we've coupled a fluorescent marker to coenzyme A on the phosphopantotheniol group. And that, because it's small, it rotates nice and quickly. If we hit that rotating molecule with polarized light, because it's rotating quickly, the light will come off depolarized. So we can monitor that depolarization. If now we've transferred this to this large protein, it's going to rotate much more slowly. when we hit that with polarized light, it's going to come off less polarized. So essentially, we're getting slow rotation, so we're going to get polarized emission, and you're getting depolarized emission with the CoA. Okay? So that's the theory. This is how we do the coupling reaction, and this is how I like chemistry to work. I'm not a chemist, don't know much about it really, but what we do here in this reaction is we take this, we mix it together in a test tube on ice, and it does this. Quite remarkable and great, and nice and easy for a biologist. And also, so we've got this fluorescently labeled CoA. We need to isolate our proteins now, so we make PPTA and AARA, amino adipate reductase, which is going to be the target we're looking at. We make that in E. coli and isolate these proteins and purify them, so we can do that quite easily. And then we build the assay. So we can see here that over time, in the absence of our enzyme, we see no increase in polarization. And as we add more enzyme, we see an increase in polarization. And this is an enzyme-dependent and a time-dependent assay, exactly what we want for a high-throughput screen as well. And we can see that we get very good reproducibility in our assay, so we get what we call a good Z-score. So the Z-score there is looking at the reproducibility of the negative result, the reproducibility of the positive result, and what we call the signal window between positivity and negativity. So now we can use this assay to look for novel antifungals. So we've been through all this process now, and we're getting to the point of trying to find out, is PPTA druggable? Crikey, we've had to do a lot of work to get to this point. If PPTA isn't druggable, what a waste of time that's been, right? So fortunately, we've identified inhibitors for PPTA. This is an IC50 curve, and we can see that we've got two compounds here that are actually quite potent inhibitors of the Aspergillus PPTAs in vitro, but not the human equivalent, so that's great, we've got selectivity. But what about against the whole cell? Great. Well, this one's not so great because it doesn't inhibit growth. But this one is fabulous, right? We're inhibiting growth. So we're saying activity against the enzyme and inhibition of growth. Now, all we need to do is prove that that's right. So if we feed these strains with iron and lysine now, that should reverse the action of the drug. And indeed it does. So great, fabulous, huge sigh of relief. We've been through all this process, we've identified a target, and it's a druggable target. So, what if we don't want to go through all that heartache of thinking, we go through all this validation process, and at the end we have to think, is this a druggable target, and only find out right at the last minute? Or what if we use an omics approach to drug discovery, apply omics in drug discovery, an alternative method? So we've got the relatively large numbers of antifungal compounds have been described, but the way in which they act, the mechanism of action, the drug target hasn't been identified in the vast majority. So is it possible to take these good quality drug targets and identify the target, these good quality drugs and identify the target? So straight away, because we know that these drugs are active, it overcomes issues with membrane, cell wall penetration. We're only working on druggable targets. And it overcomes issues with selectivity straight away, because we're only working on drugs that are active against Aspergillus, but don't show any major activity against isolated mammalian cells. So can omics technologies help? So one technology that we've heard about already is transcriptomics using next-gen sequencing. And we can look at the action of a drug against our bug and see how the bug responds. Maybe that will tell us what the drug target is. So we can do this with known drugs. So we'll take itraconazole. We know its target, CYP51A. This is the response of Aspergillus to CYP51A. These are the top 15 most upregulated genes in Aspergillus in response to itraconazole. Now, you don't need to look for CYP51A, it's not there. In fact, that's not working very well. I'll tell you right now, it's listed in around about 750th place. So if we're going to use transcriptomics to look for drug targets, it's there, look. If we're going to use transcriptomics to look for drug targets, we're probably never going to find them. And what about a novel drug? Well, very kindly, F2G, who are a drug discovery company, antifungal drug discovery company based in Manchester, have allowed us to work with one of their antifungal agents that they think has, they have indication of activity against some lipid biosynthetic target. And they've given it to us to play with our transcriptomics approach. And we can see, we get genes upregulated, we get genes downregulated. And if we look at just these upregulated genes, we can see, okay, we get a bunch that are involved in lipid metabolism. Great, okay. So that matches what the company says. But it also does a bunch of other stuff as well. So whether we'd have picked out lipid metabolism based on the transcriptomics, I doubt. But what if we compare the transcriptional response of this drug to itraconazole? If those transcriptomic profiles match, and we know that itraconazole is involved in sterol biosynthesis, if those profiles match, then that might give us an indication. So what we do is we plot the transcriptomics response of one drug against another, and our expectation, if they are doing the same thing, is that you get high responses to one drug, which is plotted across the top, and also high responses to the other. So if we got 10-fold increase here, we'd also expect to get about a 10-fold increase here. But we don't see that at all. In fact, there's very little correlation between what's going on between the two drugs. So what else can we do in terms of omics technologies? Well, chemical genomics has been used quite widely in fungal biology to identify drug targets. And I'll explain now what chemical genomics is. So if we take our cartoon cell only has two genes, a red one and a green one. This red gene produces the red proteins, which are the red blobs, and the green gene produces the green proteins, which are the green blobs. Our drug is active against the red protein. And as we increase the amount of drug binding to these red proteins, we begin to inhibit the growth of this cell. Not enough red protein around. So now, if we knock out one copy of our red gene, we've only got one copy left, but we only have half the amount of protein left. So this cell is hypersusceptible to the drug. We only need one block of drug to begin to inhibit the growth of this cell. And conversely, if we knock out the blue gene, we're going to have half the amount of blue protein, but that won't have any effect on the strain's sensitivity to drug. So if we grew these three strains together with drug, we could see this gene, this strain here, is hypersensitive. Therefore, it's probably this gene that's involved in the drug action. So can we do this on a grander scale in Aspergillus? And so this is a suite of mutants, a suite of strains, and we've tested them with itraconazole and another antifungal drug, Bromley. So, and each of these is plotted here. Each of these is a strain, and their relative growth is plotted. So these down here are the slow-growing strains, the hypersusceptible strains. And great. For itraconazole, we're identifying ERG11A as the drug target. Perfect. It's working well to identify our drug targets. And Bromley A, our target is ARF2, so that's working perfectly. But what was a bit strange to us is we identified this gene as well, ATP17, as being resistant. A bit of a surprise. Maybe it was a flaw in the experiment in what we did, but actually when we grew them out, again for Bromley, so this is one of our control strains. This one was in the middle of the pack. This is our sensitive strain, ARF2. Actually, it's not so easy to spot but actually this was a pooled analysis that we did. We used a barcode sequencing approach to look at this. So it was a DNA marker in terms of trying to distinguish between the fitness of these strains. And you can clearly see that ATP17 really is resistant. So we don't quite know why ATP17 are single knockout of genes resistant, but it's interesting. It's an interesting biological question to answer. I think I'm just running over time a little bit now. So when we look at our new drug, we can see that we're getting hypersensitivity to really two strains here that stand out, COP1 and SDA. And we can do the same sort of study in Saccharomyces cerevisiae. So that was in Aspergillus, this is in Saccharomyces cerevisiae, and we get the same result. So we can see COP1 here, and SDA, which is called OLE1 in Saccharomyces, and this is a delta-9 desaturase. So what's the link? COP1 and SDA. Well, SDA is stearic acid desaturase, delta-9 desaturase, so it converts stearic acid to oleic acid. COP1 is involved in retrograde transport from the Golgi to the ER. So no obvious link there. However, you look in the literature, and it begins to tell you the COP1 complex is a regulator of lipid homeostasis, and the ARF1 COP1 machinery acts directly on lipid droplets, which contains stearic acid. So there's a biological link there. It makes sense. That's not really where we're going with this study, but it seems to be linked together. So we go back to some of the work carried out by our partners at F2G. So this is the pathway. We've got stearic acid, oleic acid, and then linoleic acid, the next step. So if we take our drug and we don't supplement with any of these agents, so we've got 4 different drugs, 4 different analogues of the same drug. So with no fatty acids, we don't get reversal. With stearic acid here, we don't get reversal. But when we add oleic acid in, we get reversal of the drug action. So it does seem to follow that perhaps it is stearic acid desaturase that's the target of this drug. And if we look at treating Aspergillus with the drug, we can see that the ratio of stearic acid in the cell to oleic acid is affected. So we start to get more stearic acid as the drug levels increase. So again, providing evidence that SDA is essential for virulence. Is the target of the drug. And in the literature, there is a drug out there that targets stearic acid desaturase. It's called EVO86. And if we do our transcription analysis now with EVO86 and the SDA inhibitor, we can see that now we get a correlation. So the evidence builds quite strongly now that SDA is the target of this drug. So we've identified a drug target through combined approaches. Okay, so now we go on to validate the thing. The only thing we don't know is this drug target going to be essential in a mouse model? So no matter which way you work it, right at the very end, you're still waiting for the answer to the question. So we, again, this time, because if we knock out SDA, because it's essential for viability and we can't rescue it very easily, we can't work on the strain. So we couple this instead, we couple a promoter, a tetracycline inducible promoter, to our SDA gene. So if we don't feed so this is our wild type strain, if we don't feed our strain now doxycycline, so it's doxycycline inducible. If we don't feed the strain doxycycline, we don't get any growth. But as we start feeding more and more doxycycline, it's beginning to switch on SDA and rescuing the phenotype. So SDA is working in the presence of doxycycline, as we expect. So now, if we keep the gene switched off and then feed with oleic acid, you can't see it very well here, but we are reversing to some level the activity of of the knockout. So, now let's look at virulence, important factor. And yes, we can see that in all our models, so we've done a larval model, pulmonary model, and systemic model. We can see in all instances that, yeah, SDA is required for viability in our infection model. So, finally, just to sum up, I've told you about two different targets. SDA and PPTA are both critical for viability. They both have actually human homologs, but they are very different from each other. They're both druggable targets. Actually, we have an assay to monitor PPTA. SDA is a bit more difficult to work with because it's a membrane protein. So the assay isn't exactly high throughput. And we don't have any structural information yet for SDA or PPTA. And because SDA is a membrane-bound protein, it's going to be really difficult to get a structure for SDA, but PPTA we're much more hopeful of. So I really do believe that these two are valuable, druggable targets that we may see inhibitors developed for in the future. I'd just like to thank all the people, and there are a lot of people that are involved in this study, but I'd like to point out Fabio, who did all the work with SDA, generating the mutants and evaluating the strains. I'd like to thank Anna for doing a lot of the PPTA work, and Daniela for her support, and Daryl for her work on the chemical genomics. I'd really like to thank the guys at F2G for being so supportive. Mike and Jason have really been supportive of this project and supplying compound. Obviously, if you want to hear anything about F2G's new drug, Nicola Beckman is in the audience, so you might want to go and talk to her later. Okay, so thank you very much, and I'm happy to take questions.